Hi, everyone. Happy New Year. Welcome to our January 3rd Thursday, brought to you by the BC Wine Grape Council. Today, we're going to learn about some interesting vineyard technology from Dr. Simone Castellarin and crew. Dr. Castellarin is the Associate Dean of Research at the Faculty of Land and Food Systems, as well as the co-director of the Wine Research Center at UBC. His lab investigates the biological mechanisms that determine grape ripening and quality, and how these mechanisms are affected by environmental factors, temperature, and water. So welcome, Simone. Welcome back.
Thank you. Thank you very much, Kathy. Thank you very much for the invitation of sharing this research project. I will just introduce actually the speakers and the project, because this was a study that we conducted in collaboration with Christopher Mark of Vitality for the past 3 years, but was mainly conducted by Ping-Ray Chen, who is here presenting today. So I'll just give a brief introduction and introduce Ping-Ray, and then he will go through the whole presentation, and then during the question time, maybe I'll jump in again. So the title of the talk is Remote Sensing Technique to Determine Intra-Vineyard Variations in Vigor and Fruit Quality. So the project aims to understand the variability of different parameters that go from vigor to nutrient status and grape quality within vineyards. Ping-Ray will explain that this was conducted in a couple of vineyards in the Okanagan. Ping-Ray, can you move to the next slide? Yeah, so this study was originally, comes from a collaboration that started 4 years ago with Vitality and Christopher Mark. And the focus of the original collaboration was actually how to manage fertilization in a precise manner in vineyard in order to, target the plant's needs and use remote sensing techniques to assess the needs in fertilization of the vine, and also an equipment that Chris and Vitality have developed, what we call the chem box, that allows to precisely apply fertilizers in the vineyards depending on the need of the plant or soil fertilizer content. However, within this collaboration, we started conducting large surveys in vineyards of quality, like intra-vineyard quality surveys. And we thought about taking advantage of these large data sets we were taking to tackle the issues of variability within vineyard, and how we can exploit them to, in the future, potential develop strategy to reduce the variability, or as Ping-Ray will talk to, have selective harvest of grapes that have different quality levels within a vineyard. The project was, as I said, a collaboration between Vitality and us at the University of British Columbia, but was also funded by MITAX and the BC Investment Agriculture Foundation. And also was a project that was conducted in collaboration with 2 wineries, Blasted Church and Tantalus Vineyards. And I would like to thank the wineries and the vineyard managers for contributing, viticultures and vineyard managers for contributing, Scott Woody, Felix Egerer at Tantalus, John Bailey and Ben King over the years at Blasted Church, and for allowing us to enter the vineyards and contribute to that. So before I close this introduction, I just want to introduce the speaker, that is Ping-Ray Chen. And Ping-Ray is a master student, was a master student in our laboratory, worked 3 years on this project, 2.5 years. He just graduated in December. Now he's back in Taiwan, where he's originally from. Interestingly, Ping Ray is well graduated from National Taiwan University in horticulture, and he graduated from a lab in horticulture and viticulture. It is the same lab, I understand, from where Dr. Ben-Bing Chang graduated time ago. So we are very familiar with Ben-Bing and the good work he's doing. The Okanagan Ping Ray comes from the same school in Taiwan. Unfortunately, Ping Ray is moved back to Taiwan now, but I understand he will start probably soon a PhD in United States. So, well, good luck to him for this career. But today, Ping Ray, you still have some duties, and so please go ahead with your presentation.
Okay. Thank you for Simone for your introduction. Okay, so everyone, my name is Pinray Chen, and today it's really happy to share my master research work here with everyone. So as Simone mentioned in the introduction part, this is a collaboration project involving with the Health from Vitality and our two research vineyard, Blessed Church Vineyard and Tentus Vineyards. So to start with the presentation, I would like to give a brief introduction about the intra-vineyard variation. So there are a lot of variation in the vineyard, as we know. When we walk in the vineyard, we can observe the vineyard are so vary in relation to vine vigor, grape quality, and grape maturity. And where's the variation coming from? So here are a lot of sources that might be causing the variation, such as topography and soil property, soil nutrient composition, and nutrient availability for the plants and individual plant condition and vigor. For example, for the soil nutrient composition, here I give a short example of the soil nitrogen maps. So this is the soil nitrogen maps collected in one of our research vineyard. So we could observe they are as the laser point show, there's some parts have like nearly no nitrogen contents in the soil. Like here, like the value are really low, like 2 ppm in the soil. However, just like 20 or 30 meters away from this point, there are region have like 8, 9 ppm in regarding soil nitrogen. So the soil nitrogen could vary a lot in the VR, even in a really short distance in a small scale. And this variation could cause a lot of parameters to be changed, like vine vigor, yield, grape quality. So from a previous research about the vine vigor, I'm giving an example from a soil blonde vineyard. This is a research conducted in Marlborough in New Zealand. And we do notice in this map, they colored the map with a different vigor level, with red color indicated low vigor and the blue color indicated high vigor. So in this vineyard, we could notice the canopy size could be varied from one part to another part. And like as laser point showed, like the pattern is like not kind of random, and the low vigor part and the high vigor part could be really close to each other. And then I will also give a brief give an example about the grape quality. So in this another map, they conduct the research in the Shiraz vineyard in Australia. So they investigate a compound called rutodon, a secondary metabolite, which is responsible for pepper flavor, pepper aroma in Shiraz grapes. So this specific secondary metabolite have like a really very distribution in the vineyard. So here we could notice the darker color represent higher concentration in grapes, and the lighter color represent lower concentration in grapes. So we could notice some grapes, the concentration could be like as low as like below 20 nanogram per kilogram, but in some region, the value could be high, like over 600 nanogram per kilogram. And in this research, the grapes could be vary from, the Rotterdam concentration could be vary from like 70, the value could vary from 70 to more than 1,000. So it is really surprising that the secondary metabolite could be less vary even in a really short, relatively small scale research. And also, I'd like to share some nice picture in Okanagan Valley. So here I'm showing two set of picture. For the left section is the vigor one, which is the picture was took in the Tantus Vineyard in July 13, 2022, which is the growing season. So in the Riesling vineyard, we could notice in our research side, we observed some vine are really vigorous, like the left one is looked really healthy, nice. But on the other hand, on the right one, it's quite obviously the vine is not that healthy. The canopy is relatively small and it looks probably in nitrogen deficiency, which is we probably observe quite often in the vineyard. And the vigor difference might causing the grape maturity to be vary. So on the right side, I have a set of picture I took in another vineyard, viticulture vineyard in October 2nd in 2022, which is the harvest time for this vineyard. And the growing quality is Gewürztraminer. So the left one, we could observe the grape berry are, the color is turned and it's like pretty ready to be harvested, like it's red and nice. But on the other hand, the right one, as shown in the screen, the grapes is still looks like it's a bit immature and not really ready for the harvest. So it is often that we could observe this situation in a VR. And then why the intra-vineyard variation is important to the winemaker, grower, and society. So I would like to start with this part now. So for the winemaker, variation in grape quality can alter the traits of the wine. And if we understand the variation and the pattern of the grape berry, we have the possibility to produce optimal wines. So I'm showing another example from a previous research conducted in the Sauvignon Blanc vineyard and in Marlborough, New Zealand. So this research team designed a grape index, which is a calculating index based on the grape berries, sugar contents, titrable acidity, and pH, which is a composite index to describe the maturity of the grape berry. So they designed this specific juice index to make a optimum Sauvignon Blanc wine. So in the research results, they find out when the index reached to the 4.4, they have the ability to make an optimum wines. But the date for the grapes to reach that specific index have the time differences as shown in the map here. So we notice in the red region is when they reach to the juice index to 4.4, and we observe in the map on the bottom region, they already reached to the red region. However, on the other hand, on the other region, they still haven't reached to that region. So if the grower harvests all the grapes at the same time, at the same batch, they will probably not have the optimum grape quality to make the optimum wines. In this case, if they have the ability to understand the pattern, they could harvest the ratty berry, like the grape berry grown in the red region first, and wait for the grapes in other region to be ready. And also in another research, they do the similar study, which is they calculate another index to quantify if the grapes are in the mature, in the optimum quality or ultra quality. So in this Taberino vineyard research, they use a couple of parameters such as the sugar, the tetrable acidity, the pH, the anthocyanin content, and the phenolic content to calculate to as a standard to separate the two regions. And they do use this specific standard to segregate the vineyard into the premium wine vineyard region and the region that could provide ultra premium wines. So for the winemaker, if they could understand the variation and the pattern of the grape berry quality in the vineyard, they would have more ability to produce the optimum wines. And for the growers, the uniformity of vine growth could improve the efficiency of culture management. So I'm showing two common culture practice in the vineyard. So the left one is the canopy management machine. So just for the spraying system as an example, if the vine grower, sometimes they need to spray some chemical to like pesticide to avoid the disease spray. And if the vineyard could be more uniform, they will be much easy to conduct this practice. And if the uniformity is pretty low, which means that some of the region the vine is really in severe disease and some are not, they will probably either to waste too much chemical or pesticide on the healthy plants, but on the other hand, they may spray too much in the healthy plant and cause the environment issue. So a more uniform vineyard could help the grower to better control the vineyard and to make the cultural management to be performed in a more efficient manner. And for society and environment, they could reduce the risk of contaminants by excess chemicals runoff, especially for the vineyard. The vineyards usually located near to the large water body, such as lake or the river. Much unnecessary chemical, they will be just contaminate the water and they go into the lake or the river and contaminate the water resources. So concerning this kind of problem and situation, a novel idea comes out, which is the precision viticulture management. So the precision viticulture management could be in the several concepts, such as a variable rate application and zone delineation and the selective harvesting. For the variable rate application, I would like to use one of my research project as an example. So we conduct a variable rate fertilization in our projects. So this kind of technique could apply the different doses of the fertilizers to they change the value of the fertilizer within the vineyard. So the fertilization could be conducted in the vine specific manners. In this case, we consider the individual vine's condition and individual vine's needs. And in this case, we could avoid wasting the fertilizers. And the other one is zone delineation, and the selective harvest is within this concept. So the basic concept for the selective harvest is the grower could harvest could separate the grapes quality and harvest at a different times. For example, in one of the study I mentioned previously in the Taberno vineyard, they could harvest the region of premium wines grape and ultra premium wines grapes separately to produce the different quality of wines and sell them in the different prices. And also a research study conducted before already mentioned, they do the profits analysis, and they do mention the selective harvest could be profitable and manageable. Okay, and since we are interested in the grapes aroma, which is the one of the major components in the Riesling and Gewürztraminer wine in my study. So Riesling and Gewürztraminer are two important white grapes cultivar in the Okanagan Valley in Canada, and the unique and strong aroma content is they are characterized by their aroma. And for these two aromatic cultivar, terpenoids are major contributors. And the terpenoids are usually present in really small concentration, usually in the unit of microgram per kilogram, but they could have a tremendous impact on the overall quality. And the terpenoids are usually responsible for the floral and the citrus aromas, and the profile could be variety dependent. For example, in the Riesling vineyard, we usually perceive strong floral and the citrus aroma, which is mainly caused by the Riesling high concentration of terpenoids. And then I would like to talk a little bit more on terpenoids. So they could be extremely diverse and estimated to comprise over 40,000 individual compounds. And the form could be monoterpenoids and the sesquiterpenoid. These two forms are usually the two free, or often we call it volatile form for the terpenoids. And on the screen, I'm pointing a few common terpenoids usually give the aromas. Limonene and terpene, they have the aroma of the lime flavor and the lemon flavor, and the lialo usually have the floral and lavender flavor, and the Nero and Gerano often have the rose and lychee flavor. And the concentration of terpenoid in grapes could be affected by biology factors and also cultural practices. The previous research have pointed out if by the leaf removal practice and to allow more light exposure to the grapes, the concentration of terpene could be enhanced. And also another research point out the decrease of crop load by cluster seeding could also enhance the terpenoid concentration in grapes. And next, I would like to use a couple of slides to describe my material methods. So first, I would like to talk about the experimental side. So the research is conducted. One is BCWGC vineyard in the Okanagan Falls, which is here near to Penticton, and the grow cultivar is Gewürztraminer. And the second vineyard is Tantus vineyard, which is located in Kelowna, and the grown cultivar is Riesling. And as I described before, these two cultivars are considered aromatic cultivar due to their high concentration of terpenoids. And this is the slides to talk about the experimental design and how we collected the data. So in the BCWGC vineyard, the research is conducted in 2022 and 2023, and the research region is around 1 hectare. And in this 1 hectare field, we divided them into 48 plots as shown in the figure here. And in each plot, there are 28 vines per plot, and we randomly selected 3 vines per plot to conduct the data collection and the sample collection. And in the Tantus vineyard, the ground cultivar Riesling, and we have 3 years data for these vineyards. And the research area are also around 1 hectare, and here we have 40 plots in this vineyard, and we have 50 vines per plot. And similar as BCWGC vineyard, we collected the data and a sample from 3 random vines per plot. And there are quite a few data we collected in my study. So the first part is plant physiological analysis, which I collected this kind of data in both blooming and virgin time. So the first one is water potential. We collected using pressure chamber and to assess the water status of the vines. And the second one is leaf gas exchange using a portable gas exchange system, which we could understand the photosynthesis rate, transpiration rate, and the stomata conductance of the vines. And also we calculate the leaf area using the leaf area meter and to understand the canopy size of vine in blooming and virgin time. And also interesting, we also collect the leaf spectroscopy data using ground measurement with handheld spectrometer, like the figure here. And also the vitalities help us to collect the drone data using drone to collect the remote sensing based leaf spectroscopy with using augmented aerial vehicle equipped with a multi-spectrum camera. And I would like to introduce about the leaf spectroscopy data. So to assess the vine vigor and vine health, two vegetation index was developed. And these are the most common vegetation index. So the first one is normalized difference vegetation index, NDVI. And the second one is red edge normalized difference vegetation index, NDRE. So the calculation formula are pretty similar. The only difference for these two vegetation index is their wavelength used to calculate. So they calculate based on the reflectance of the specific weightage, and to calculate a single value to assess the vine vigor. Use this range to separate the vine vigor. So if the value is ranging from, this one is for NDVI. So if the value ranging from 0 to 0.3 is usually considered as unhealthy plants. And if it's the value is ranging from 0.3 to 0.6, the vine was considered a moderately healthy. And if the value is in a range of 0.6 to 1, it usually considered as healthy plants. And this is an example from the drone image and a DVI value. So here the red color represent the higher value, with the maximum of 1, and the blue color represent the lower value. So here the red color represent here is this is a house and the row, which is not the vegetation. And in the VR we could notice there are some parts have the more reddish color, which indicates the vine here is more vigorous, and on some part, like here, the color is more greenish, which indicates the vine vigor is a little bit lower. So in the VR, the vigor could be varied. And also I conduct other measurements. One of important one is the plant nutrition analysis, which is using the patio analysis collecting in variation. And nutrient profile was obtained, including nitrogen, phosphorus, and potassium and boron. And also for sure, the grape quality analysis was included in the projects, and we used the harvest grapes. And some basic technological parameter was collected, including sugar content, tetrabric acidity, pH. And for this project, we collect water terpenoid using the SPNE GCMS to accurately quantify the volatile terpenoids concentration, which include 24 individual terpenoids. So the objective for this study is to assess the spatial version of vine-related parameters and the volatile terpenoids, or we can call aroma within vineyards, and to identify vine-related parameters that correlate with volatile terpenoids. And since as in the project, we noticed the volatile terpenoids is important to the quality of Riesling and Gewürztraminer, but it is really labors. It requires a lot of labors and money and time to conduct this sort of analysis. So we are trying to develop a remote sensing strategy to estimate volatile terpenoids level within vineyards. So here's come to the interesting and exciting part, which is the result. So here, I would like to share the result of intra-vineyard variation in the Gewürztraminer vineyard. So here we can see four distribution map, and the map was colored in 5 different colors, which indicate the different range of the value, with the red color indicate the higher value, orange bar, blue color indicate the lower value. So pretty much the data was segregated into these 5 color regions, and which indicate in each region they pretty much have the same number of data points. In these methods, we could understand the distribution of the value for the parameters. For example, in the leaf area, we could notice a higher leaf area was observed in the middle part of the vineyard and the bottom part of the vineyard, as the laser point showed. And the lower leaf area region was observed in the upper part of the map on the both sides, like here and here. And I used the same methods to color the map for leaf area and DRE, one of the vegetation index we mentioned previously, and also yield and total free terpenoids concentration. So when we draw the map and do the data analysis, so we found some interesting things, which is this, for example, for this map, this map, the distribution is not exactly the same, but they do share the similar pattern, and it seems like these parameters are probably correlated together. So to verify their correlation, I conduct the correlation analysis. So in the correlation analysis, we do the statistical analysis to understand the correlationship between the parameters. And the red color indicates the positive correlation, and the value within the red dot or the dots indicate the strength of the correlation, with higher value indicate higher strength of the correlation. And the red dot, the reddish color indicate positive correlation, and the blue dot indicate a negative correlation. And with the crossing part is indicate the correlation is not significant. So we collect a lot of data, but to simplify the data and to better interpret the data, we choose a few parameters and separate them into 4 sections. So the first one is the vegetative indices, which I conducted, including the NDVI value from ground measurement and remote sensing based measurement, and also the NDR value from collected from ground measurement and remote sensing based measurements. And also for the wine physiology, I include the net assimilation rate, which also refers to the photosynthesis rate, and also the leaf area here. And for the new phytonutrition status, I including the nitrogen and phosphorus and potassium. And the last part is the productivity and the berry quality, which I include the yield and total free terpenoid here. So there are a lot of correlation here. We do observe some interesting findings, but I would like to have your attention on the first part. It's the correlation between vegetation index and other parameters. So here, as we do observe the positive correlation between vegetation index and the leaf area, like here, and the nitrogen contents, yield, and total free terpenoids. And also we observe vegetative index VI negatively correlates to phosphorus content. And next, for the total free terpenoids, TFT, we observed the positive correlation between TFT and vegetative index, like here, and also the nitrogen contents and yield. And we also observed the negative correlation between the total free terpenoids with photosynthesis rate and phosphorus contents. So it's really interesting to find out there are some correlation between the vegetative index and the total free terpenoid and other parameters, but it's really hard to apply this kind of knowledge to correlation in the practical production system. So I conducted another study, which is the principal component analysis study, PCA study. So in this study, in this kind of analysis, we could turn the correlation into a more easy visualized format. So in this graph, there are a lot of arrows here. So when the arrow pointed at the same direction, they means these parameters are correlated positively. For example, for the green circle one, the NDRE, remote sensing-based NDRE, and the purple circle one, the total free terpenoids, the arrow are pointed together, which means they have the strong positive correlation. And the dots here was indicate that all the data points collected in the data. So I separate the data points into the low vigor group and the medium vigor group indicated by blue color and the red colors respectively, based on the remote sensing based NDRE value. So here we could observe a medium vigor group is pointed on the right side, and the low vigor group is on the left side, which we could observe these two vigor groups are separately, and it seems like a medium vigor group is more closely to the total free terpenoids concentration, which means it's likely medium vigor group have more total free terpenoids and probably have more nitrogen contents than the low vigor groups. Therefore, I conducted the further statistical analysis to verify this kind of gas. So first, for the field parameter in the Gewürztraminer, here I show the data in two different formats. So the blue color indicate the value of low vigor group, and the red bar indicate the value of medium vigor group. So here in the Gewürztraminer, we notice for the leaf area, although there's a trend that medium vigor group have higher leaf area than the low vigor group, but the value are not significant different. But if we move on to the nitrogen part, we do notice medium vigor group have higher nitrogen level than the low vigor group. And in the yield part, we observe the similar things, which is medium vigor group have higher yield than the low vigor group. And also interested in the total free terpenoids, we also observe medium vigor group have higher aroma concentration than the low vigor group, which is a little bit out of our expectation, because in our previous collected papers and knowledge, it is said that the higher yield, higher vigor might reduce the grape quality, and we in our assumption, we thought the medium vigor group will have lower total free terpenoids. But in the end, in the research study, we do notice actually in the medium vigor group, they have the higher total free terpenoids concentration than the low vigor groups. And then let's move on to another vineyard, Riesling vineyard. So we observed the intrinsic findings in the Gewürztraminer in that 2-year study. So we want to know if we could see the similar results in another cultivar in another site, to see if the correlation is just random, or it could be like robust across the in different cultivar. So here the masses to generate map is same as the previous one, which I separate the data point into the 5 groups and color in the different color, with red color indicate higher value and the blue color indicate the lower value. So in this format, leaf area map, NDRE, yield, and total terpenoids, we observe similar. The pattern are not exactly the same, but we do observe the similar pattern, which suggests that they might have some correlation between the parameters. So similarly, I conduct the correlation analysis in this Riesling vineyard, and the parameter I chose is same as the previous, so I would just skip this part and go straight to the results part. So the vegetation index positively correlates to the photosynthesis rate, leaf area, nitrogen contents, yield, and total free terpenoids, and also it negatively correlates to phosphorus contents. Which is the result are pretty much similar as the previous Gewürztraminer and vineyards. And also in the total free terpenoids, it positively correlates to the vegetative index, photosynthesis rate, leaf area, nitrogen contents, and yield. And it also negatively correlates to phosphorus content. So it's really interesting that we find really similar results between these two data sets. And also I conducted a PCA study, which I explained before. So here we also observe the total free terpenoid and NDRE, remote sensing based NDRE. Their arrow are pointing at the same direction, which refer to they have the positive correlation. And also the data point here, the dot here indicate the each data point, which I also separately them into the low vigor group and the medium vigor group, which we could observe 2 groups are quite separated, which indicates it's a nice separation, and we might observe the significant difference. They might have the different property of these 2 groups. So in the leaf area part, in the 3-year study, we observed the medium group have higher leaf area in each year, which is pretty interesting. The data is stable. The value are pretty much stable. And for the nitrogen, we also observe the medium vigor group have higher nitrogen level than low vigor groups, as shown as the data here. And also for the yield, we also observe medium vigor group have higher yield than low vigor group. One thing to note is we can notice the value are pretty low in 2023, which is because of the severe winter damage happened in the winter of 2022, which damaged the bud. And as you know, the production is pretty bad in this year, the 2023. But even though in this case we do observe the difference still be observed in the medium vigor group and low vigor group, which indicate the result are still be stable. And also for the total free terpenoid concentration, we do observe the similar things, which is medium vigor group have higher total free terpenoid concentration. So here is the concentration and the future direction plot. So we observe there are large intra-vineyard variation of volatile terpenoids, or we can call aroma level within the vineyards. And also volatile terpenoids positively correlates to vegetative index, such as NDVI and NDRE, and also nitrogen contents and yield. And in the future, in the further static analysis, we determine that low vigor, determined with remote sensing based vegetation index, has a lower yield and lower aromas. And also, based on the study, we also confirm that remote sensing based vegetation indices can be used to separate vineyard plots based on volatile terpenoids level. which means in this study, we could separate the grapes with higher aroma content and lower aroma content. And if we perform the further zonal management, such as selective harvest, we could have the different, we can have grapes of different aroma contents, which probably we could, in the future, we could produce the wine of like really strong aroma flavor and regular aroma flavor wines. And this is the possible precision measurement workflow diagram for this study. So first step, we do observe their intra-vineyard variability regarding the vine vigor and also for the grape quality. And in the growing season, we could collect the UAV best image. As shown here, we collect the data, and after the process of noise removal and the vegetation index calculation to turn the data to another format, and we do the statistical analysis such as cluster analysis to separate the plot into the high quality region and the low quality region. And the grower could conduct the management, which is how we could harvest the grapes separately in a different batches, which have the premium quality grapes and the medium quality grapes. And in the end, we could probably increase the efficiency and maximum the benefits of the grapes. So in the end, I would like to appreciate my help from my collaborator, Christopher Mark, and the Vitality Technology Incorporation, and also the supervisor community member, and also the lab member in our lab to support me with the data collection in the field. And also, I would like to appreciate my funding agency, Vitality, and the two research vineyard, Tentus Vineyard and Blessed Church Vineyard. And thanks for listening, and I will happy to take any question.
Thanks, Penry. That was really interesting and a little surprising, right? Because I think that the prevailing thought is that the lower your yield, the higher your quality, right? And
yeah, that's true.
And so I think this could make a lot of changes in vineyard management and encouragement of healthy vines, right, rather than stressed vines.
Kathy, can I comment on these just for a second?
Please, yeah.
I agree with you. It was a surprising data for us as well, and was consistent across the year. But I would like to comment on one thing, and that's also the reason why we call medium vigor and not high vigor. As you can see also from these pictures, I think, being Rafe, correct me if I'm wrong, this is one of our experimental sites, right? So we are not talking to very high levels of vigor, and we were not talking about very high levels of yield. So we are talking on numbers that you have seen, for instance, 2 kilograms per vine, 3 kilos per vine. So often I think when we infer about high crops and high vigor, we are in some studies, in other studies, they were very much higher, what I want to say, much higher crop levels and much higher levels of vigor of the plant. So yes, it is surprising. I agree, because we were expecting to anyways observe a trend toward more quality with less vigor. But we also want to acknowledge that these vineyards were in a good shape. They were optimal vineyards where somehow where you know we those are the commercial, I would say, commercial vineyards in the Okanagan, where we don't overcrop and we tend to have contain vigor in our plants. I just want to clarify that you know, we are talking about medium vigor because we think it's a good vigor level, is not like an extreme vigor level, and etc.
Right. Did you think of looking at berry size as a parameter?
I think we do have that value. We did not include that in the correlation map that we show because for simplicity. But Pinray, correct me if I'm wrong. And we did not observe. Did we observe any correlation with berry size? I don't think we observe a correlation with berry size, did we?
No, for the berry size, the correlation are usually weak.
Yeah.
Not that strength, and yeah, and it also remind me one thing. So also we collect the sugar content, and which I didn't show the data here in the correlation map, which is basic because we do observe the sugar contents don't have really significant relationship with other parameters, which also the sugar content don't have the correlation between didn't correlate to the total free terpenoids, which also indicate it's the sugar may not be a good predictor for the aromas.
Yeah, that was another thank you, Ping-Ray, for mentioning this. This was another thing that we were expecting, actually, that there is not a direct relationship between sugars and terpenes, and we didn't show this graph, but also about the variation within a vineyard in sugar was not as high as the variation we find for terpenes. So that is also consistent with this result that sugar is not too informative for determining the level of aroma in these vineyards. But again, we are not talking about unripe fruit of 12 bricks versus fruit of 22 bricks. We are talking at harvest time, fruit that is overall ripe, but the pockets of the vineyard with higher volatiles, higher aromas were not necessarily, did not have significant difference in sugar levels.
Do you anticipate doing similar work with a red variety?
So who knows? You know, it will depend of how we follow up with these studies, and if we have we receive, you know, we are able to get new funding for it. I must say that, correct me again, Ping-Ray, if I'm wrong, because actually I would like to remind everybody, I didn't say at the beginning, this was a completely new research stream for our lab in terms of remote sensing and this type of precision viticulture techniques. And I should thank again Vintality for supporting us in all the drone and data analysis, and Ping-Ray, who came here and pretty much came up with all these analyses by himself, because in the lab we didn't have that expertise. So it's a new thing for us. I must say that related to your question is, I think they've been doing some work, maybe more work on color and pigmentation than for aroma, you know, in other parts of the world. But certainly something that actually will be even probably, I'm not sure if we'll find the same correlations, but for us analyzing color is even easier than analyzing aroma. So it's potentially something that can be done.
Right. I'm sure you said were the spectral analysis done at Verizon? Is that?
So the spectroscopy data was collected in both blooming and virgin time, like for both the ground measurements and draw measurement. It collected in both phenological stages.
What was there any attempt at the wineries to separate and ferment separately the low vigor, medium vigor?
In this project, you mean, Kathy?
Yeah.
No, actually, this is almost a secret project in the sense that we use data we were collecting also for the other part of the project, that is the variable rate fertilization, and we decided to reprocess the data to infer this, to look for this new technique or implement this potential technique. So what I'm saying is not it's a secret, but we did not. It was not meant to be a lead to having the wineries then using it within this project. But actually, that's the pleasant surprise we had, that we believe that it worked quite well. This technique worked quite well, and as Ping-Ray has mentioned, potentially it would be interesting to use it and produce wine out of different parts of the vineyard, like they've done in those other studies, and have, I don't know, a premium and ultra premium. I like that they call it, you know, this way. So and see how if it actually results in different quality of the wine.
Right. Yeah. Any other questions from the floor? Or
Just one quick comment, Kathy, while people come with questions. One of the things that Pingray mentioned, but was really interesting to me, and one of the minor pieces, but that was surprising, was how strongly phosphorus content, higher phosphorus content, was correlated with lower aroma, lower vigor, lower yield. So it was interesting, all 3 years and both sites, it was very consistent that higher phosphorus content was unquestionably a negative, and effectively, and I'm obviously being a little bit crude here, everything we studied in the study. So that was also, again, a minor piece of what we looked at, but was pretty strong and definitely surprising.
Like, were you looking for that? Were you looking at that piece, or it just jumped out at you?
It just jumped out. It was not, I mean, we were testing all these variables, but we
Right.
weren't, it was not something we expected at all.
Right.
At least I didn't. And I would guess probably the same for Simone and Pingray. It just was something we just, when Pingray did the analysis, it's something we consistently saw every year. And again, a pretty strong correlation in general. And so it was definitely a bit of a surprise.
And that's just an effect of the variation in the soil, right?
Yeah, so I would like to comment on this opinion. Yeah, it's really interesting finding in this project that the phosphorus data in vine is directly correlated to the vine vigor, and also for the totally terminal, which we didn't expect at the very beginning. And actually in the result for the phosphorus contents it's in the vines we collected, the phosphorus content is six the recommended contents. So there's a specific range for the phosphorus contents in the vines, and across the two research vineyard and in pretty much all the data points the sample collect, we observe the phosphorus have like really high values in these vines, which is have like it says the recommended values, which we guess it might be the reason why the phosphorus is actually correlates to the vigor index and also the total free terpenoid concentration.
Wow. Yeah,
Vice versa, we observe kind of medium low values of nitrogen. That was another thing that stood out. I mean, but that was also part of the strategy of the vineyards that they want to contain with nitrogen, etc. So it's they are not over fertilized. So those are commercial conditions, but those were some observation from the petiole analysis, the high level of phosphorus and relatively low level of nitrogen.
Yeah, it's known that phosphorus and nitrate are antagonistic.
Yeah.
So high phosphorus in plants can reduce your availability of nitrogen. It also can reduce your availability of some micronutrients that could be somehow involved in the processes that are affecting your terpene accumulation.
Well, thank you very
Thanks,
much, Lisa. Yeah.
And my recollection, again, correct me if I'm wrong, guys, is that also the soil phosphorus was not excessively high. Like the plant tissue phosphorus was, fairly high, but the soil was kind of typically roughly within range. And so again, that's certainly my recollection. Like it wasn't like we were dealing with really high soil phosphorus in both cases, but we were kind of dealing with what I would say is pretty standard Okanagan phosphorus levels in the soil.
Interesting.
So Kathy, don't ask me why we have, maybe Lisa knows why we have so much in the plant.
I sometimes see that in sap analysis, and it could be a function too of your soil microbiology, that you have to take phosphorus as Chris knows this, soil reports you have to sometimes take with a grain of salt and tissue. But phosphorus will sometimes see higher phosphorus in sap analysis compared to what you would expect from the soil. And it could be a function of quite efficient soil microbiology going on, potentially, that is allowing the uptake of what phosphorus is there. It's helping mobilize it, potentially. Likewise, if there's, I don't know what the sulfur levels are like in the soil there. In general, our sulfur levels are pretty low. But if you have adequate sulfur where your phosphorus is by the roots, that can also help your uptake.
Well, this suggests a whole new research project.
I do a lot of sap analysis, and it's very fascinating
Yeah.
to get out of that, and being able to evaluate plant stresses, whether irrigation or dust, for example, root issues. It's really fascinating stuff to help identify early issues.
Yeah. Yeah. Okay, we're at the end of our time. Thanks so much, Pinray and Simone. Super interesting. If anybody has questions, you can contact Kate and she will give you the contact information for Pinray and Simone, and you can fire them off. The recording will be up shortly. And February is our third Thursday is February 20th, and Dr. Ben-Min Chang will report on the survey. And Renee, yes, we will also put Lisa Wombold's contact information up. Thanks again, and we'll see you next time.
Thank you, and particularly thank to Ping Ray, who woke up at 3 a.m. to give this presentation in Taiwan. So thank you, Ping Ray. You can go to bed now.
And
Okay.
and it's so cool that the Northern Lights are going on in Taiwan right now.
Exactly. All right.
Thank you.
Hey.
Thank you very much.